Pneumocystis kirani is from Epicomplexa and it is opportunistic pathogen. It's still in the discussion whether it is a yeast-like fungus or protozoa. P. kirani is a protozoan parasite found in the lungs of the various mammalian, including the man, existing to two forms, trophozoite and cyst. The cysts have 8 intracystic bodies and expirated in the sputum. Inhalation of other susceptible hosts and sporozoites are released into the alveolar spaces, grow into the trophozoite, and it's enter into the merogyny. Some of the merozoites continue the merogyny cycle while other are involved in the cyst formation. It is reported from all over the world that infection in the man is by the inhalation of cyst, and most asymptomatic unless the profound T helper cell diffusion or depression or the P. kirani is opportunistic protozoan causing the pneumonia. It is atypical feature of this parasite is to form the honeycomb-like alveolar mask containing the large number of parasites. In the James stand, this honeycomb mask shape can be seen. And for the diagnosis, we use the glomeruli Gimsa fluorescent label antibodies and toluidine blue stain may be used to identify the P. kirani. The diagnosis can also be made by detection of parasite in the sputum, lungs, aspirate, bronchial wash, lung biopsy, and culture in tissue. Leishmania species are also from Epicomplexa. It have also called the monoflagellate parasite and reticular endothelial system. This parasite have 2 stages. What is promastigote and other is amastigote. In the promastigote there is a flagella. You can find the kinetoplast and the nucleus, but in amastigote you can find only the nucleus and kinetoplast, no flagella. Wide amastigote form are mostly found in the vertebrate host, that is humans, and the promastigote form with single flagella is mostly found in the insect host sandfly of a genus Phlebotomus or Lutzoma. In the infection is caused in the man by the bite of a sandfly and blood transfusion. Visceral leishmaniasis, scala or cutaneous leishmaniasis or oriental sore is caused by the Leishmania donoi complex and Leishmania tropica complex. And the monocontagious leishmaniasis, epodia ota and cherola ulcers are caused by the Leishmania brasilensis complex and Leishmania mexica complex. Of the only Leishmania donoi complex and tropical complex are found in the old world. The cutaneous leishmaniasis is caused by the L. tropica complex, can be both dry and wet type, and the L. donoi complex is more common in the Asia. Infection occurring in the man through the sandfly bite and kala azar is characterized by the skin depigmentation, fever, splenohepatomegaly, and anemia, hyperglycemilubinemia, and emaciation. In the India, post-kala azar dermal leishmaniasis also occur. About 20% of the cases of visceral leishmaniasis diagnosis is made by detection of LD bodies, bone marrow, LD bodies in the bone marrow, spleen, liver, and the blood smear. We can culture it into the triple N media, which is Nicholas Novi McNeil medium, or by detecting of antibodies in the serum LD bodies, Leishmania donoi. Another organism just like the Leishmania donoi is a Trypanosoma species. Trap2 means boring and soma means bodies, and this organism have also kinetoplastida, and it's from Sarcomastigiophora. And this is monoflagellate parasite distributed in West Africa and South of America. And this structure, the only addition are undulating bodies which differentiated from the Leishmania species. Otherwise this organism have also flagella, kinetoplast, and the only addition is undulating bodies. Trypanosoma are monoflagellate. Trypanosoma cruzi found in the South America is transmitted by radioactive bug, kissing bug, while the Trypanosoma rhodonensis are Gimbies are present in the West Africa are transmitted through the Glossovia species. If you see the Trypanosoma rhodonensis and Trypanosoma burosi are West Africa and transmitted through the tasty flies, which is Glossova species, and they cause the sleeping sickness, and Tripto-amastigote form are there. T. cruzi in the South America transmitted through the radioactive bug, kissing bug, assassin bug, and trimetron and cone nose bug, and cause the Chagas disease. The diagnosis is made by detecting the parasite into the blood, bone marrow, CSF serological method, and xeno diagnosis Chagas disease, in which the organism, a susceptible organism is exposed, or tissue cells are exposed to the parasite, and then check for the changes, any changes in the normal cells. Tissue in the blood, Helminthes, Washiria, Bancrofti are Burgia milai are mostly caused by the Aedes. Its helminthes and Burga timuri is also involved in the Mycophilaria group. If you see, this organism have definitive host, which is man, and intermediate hosts are mosquitoes, Anopheles, Culex, Aedes, Mansoni. The female is longer than male, and Vaucheria and the adult worm live in the major lymphatic duct of the human and are tightly coiled into the nodular masses. Vishila bancrofti microfilaria have marked actually Vishila bancrofti do not show the nocturnal periodicity. It's the only property of the Burgia milai that show the